Now, the drug absorption is a pharmacokinetic parameter determined by the drug's physicochemical properties. Now, what are those physicochemical properties? They include the solubility, the dissociation constant, partition coefficient, drug ionization or pKa of a drug, and protein binding. Now, other factors include the formulation and the route of administration of a drug. Now, let's talk about the formulation effect. Now, the dosage forms, for example, tablets, capsules, and solutions consist of the drug plus excipients, are formulated to be given by various routes, for example, oral, buccal, sublingual, rectal, parenteral, topical, and inhalational. So these all formulations are designed in a particular way using different excipients to facilitate their absorption across different parts of the body, for example, oral cavity, rectal route, injectable routes, topical routes, and inhalational routes such as delivery of a drug into the lungs. The example could be general anesthesia and the use of certain anti-asthmatic drugs such as bronchodilators, which deliver drug directly into the lungs. Now let's see what is the impact of route of administration on the drug absorption. Regardless of the route of administration, drugs must be in solution form to be absorbed. Thus, solid dosage forms, for example tablets, must be able to disintegrate and deaggregate before they are available for absorption across different parts of the GIT. Now let's see what happens to a drug when it is delivered into the body by oral route. The drug is administered via oral route. It is disintegrated and then dissolved. The absorption takes place either from stomach or small intestine. The distribution of the drug takes place by the circulatory system or the bloodstream. The drug reaches the target site or the site of action, and it produces the pharmacological effect, which could be either a therapeutic effect or a toxic effect or side effect. Now let's switch back and see what happens after distribution. So in case of a very polar drug or highly water-soluble drug, the drug may get into the major excretory organ or the kidney and then eliminated. Or it may go into the liver first via portal circulation and then made sufficiently water-soluble to be eliminated via kidneys. Now this pharmacokinetic phase is not as simple as you might be thinking. The unmetabolized fraction of the drug can redistribute back into the blood and may be available at the target site to show therapeutic response. Now this phase of the drug shown in blue could be regarded as the pharmaceutical phase because all these parameters including disintegration and dissolution of the drug are controlled outside of the body or in vitro. And this is all done by a special pharmaceutical designed for a drug to provide a predefined and predictable disintegration and dissolution pattern to the dosage form. So this is the pharmacokinetic phase, all these green ones. The pharmacodynamic phase, where the drug is in action and it is producing its own response. Now for an orally administered drug, for example tablet or capsules or a syrup, the drug absorption takes place from stomach and small intestine to portal circulation first. In case of perrectal or rectal route, the absorption takes place straight into the systemic circulation. In case of intramuscular route, the absorption takes place from muscles to systemic circulation. So in order to be absorbed, the drug needs crossing cell membranes in different parts of the body. Let's have a look here. We have an oral dosage form and an injectable dosage form. Let's talk about the oral dosage form first, which is a capsule here. So when it is taken orally, it enters into the portal circulation, which takes the drug into the liver first, which then delivers it into the systemic circulation, and then it reaches the rest of the body. So drugs administered orally are first exposed to the liver and may be extensively metabolized before reaching the rest of the body. And this phenomena is known as first pass effect, when the drug has to pass through the liver first before entering the systemic circulation and reaching the target site or rest of the body. In case of an IV injection, which places the entire drug into the systemic circulation, bypassing the liver, the situation is entirely different. The drugs administered IV enter directly into the systemic circulation and have direct access to the rest of the body. Therefore, it can be used in an emergency condition where you need a quick action of the drug. So, no absorption is required if the drug is given by IV route. As you have just seen, the entire dose was delivered into the systemic circulation. In case of oral dosage forms, the drug has to pass through various biological membranes first, the membranes of the GIT first, and then the liver, and finally they gain access to the systemic circulation and rest of the body. So from your basic pharmacology knowledge, only a non-ionized or non-polar drug diffuses across the membrane, hence this is an important factor. Non-polar drugs are lipid soluble, that's the basic concept. Polar drugs are water soluble, they can't absorb across the biological membrane. Let's have a look. So here on this side of the cell membrane you have a lipid soluble drug. So a lipid soluble drug or a non-polar drug can pass through this lipid bilayer, which is the cell membrane. So lipid favors the lipid. This could be an excellent example of nepotism. On the other hand, in case of ionized drug, which is water soluble, it will have resistance and it won't be able to pass through the membrane. Because the membrane is lipid, the molecule is water soluble, so it is not allowing it to pass through. For a more detailed concept on this, you may watch my another video. The link is given in the description. Now let's discuss a scenario. Now these are the three different drug molecules, the blue ones, the yellow ones, and the red. And this is your lipid bilayer or the cell membrane. Now as you know, the most frequently available way of drug absorption is the simple diffusion. Now in simple diffusion, the drug is transported through the lipid bilayer from a higher concentration to a lower concentration, means along the concentration gradient, not against the concentration gradient, which is the active transport, if you remember. Now if a molecule or a drug is hydrophilic or water soluble or polar, the hydrophilic drugs have a difficult time crossing through the plasma membrane. You all know that by now. If a drug is lipophilic or non-polar, it will be the most desirable to be transported across the lipid bilayer. As you can see here, it is readily penetrating the lipid bilayer, concentrating inside and then leaving out efficiently. On the other hand, there is a third drug which is very lipophilic or highly lipid soluble drug. So as you can see, the drug is concentrating within the lipid bilayer because it loves the lipid too much. Because of its exceeding love for the fat, it is concentrating within the fat and is refusing to leave out. So such a drug, which is very lipophilic in nature, may fail to pass through because the molecule is extremely lipophilic and thus will have hard time leaving the lipid membrane. Now let's have a look at the different means of drug absorption across the biological membrane or the plasma membrane. The cell membranes are composed primarily of a bilayer, and you all know that, which determines the membrane permeability characteristics. Drugs may cross cell membrane by different ways. For example, passive transport, which includes diffusion and facilitated diffusion, active transport, and pinocytosis. Now from this slide we will have a quick look at the different ways of drug transport across the membrane. The first one, as you know, is a passive transport, which occurs predominantly in the body, and it includes both diffusion and facilitated diffusion. Now on this side of the cell mem there is high concentration of the solute or the drug molecules. On this side there is low concentration of the drug. This is your lipid bilayer, and the drug molecules are happily transported from a region of higher concentration to a region of lower concentration along the concentration gradient. Let's have a look at the facilitated diffusion, where the drug molecules are transported from a higher concentration to a lower concentration. This is the higher concentration, this is the lower concentration of the drug, but here there is a specialized carrier protein in the cell membrane which is doing all this job. So facilitated diffusion is exactly the diffusion, but the only additional thing is the presence of a carrier protein which delivers the drug across the membrane from a higher concentration to a lower concentration. Let's talk about the active transport. Now in case of active transport, like facilitated diffusion, there is a carrier protein, but additionally there is one ATP molecule or energy which is driving the process even more efficiently. Now this ATP or energy is basically required to drive the drug molecules from a region of low concentration to the region of higher concentration, unlike facilitated diffusion, which is a process from higher to lower concentration in the presence of a carrier. So in facilitated diffusion there is a carrier, in active transport there is a carrier. This is the one similarity. The dissimilarity is that in active transport there is movement from lower to higher concentration, whereas in facilitated diffusion there is movement from higher to lower concentration. And of course there is one more dissimilarity, as I already mentioned. There is ATP in active transport. There is no ATP in facilitated diffusion because that is not required. The ATP is only required to drive from lower to higher concentration, means to drive the car uphill. When you are down the hill from higher to lower concentration, no ATP is required. I'm sorry, on this side of the frame the same process is taking place, from lower to higher concentration, driven by ATP in the presence of a carrier protein in the cell membrane. Let's see what happens in pinocytosis. Now the pinocytosis is also called drinking of cell. Now here the drug molecules will come across the cell membrane. It's coming close to the cell membrane, touches the cell membrane. The cell membrane invaginates. Wait for it. It invaginates and folds inside and makes pseudopods, or the pinocytic vesicles. These are the pinocytic vesicles, which then release the drug into the cell. So this is the topic for today. I'll see you again soon. Bye bye.